having started from the patients and modeled the disease and understood potential candidate therapeutic targets, the dream would be to develop a targeted therapeutic that then we do see efficacy in clinical trial.
Hey there, I'm Luca Fusar-Bassini. I'm a PhD student in computational biology at EPFL in Switzerland, and you're listening to The Biotech Futurist. The Biotech Futurist aims to foster deep understanding and discussion about exciting hot topics in biotech, but I want to say from the beginning that it is by no means rigorous in teaching the subject. And for the sake of outreach, sometimes we need generalizations that, of course, simplify the reality of the science behind what we're discussing. But I can say that my guests and I do our best to be clear and to go in depth. You can imagine to be out with me and my expert guest for a friendly conversation to get a general understanding and more curiosity, having fun as much as I've had recording this podcast. This podcast has no sponsors and any reference is not meant to support any commercial activity. This podcast is a solo effort, so if you wish to support me, I'd be grateful if you followed The Biotech Futurist on Spotify, Apple Podcasts, YouTube, Instagram or your top podcasting platform and share it with your friends. With that said, I am excited to move on to today's conversation at The Biotech Futurist. Today, I am really excited to interview Christina Theodoris. Christina is a visual artist striving for rhythm, balance and compassion, with a long artistic story including exhibitions and awards all over the US. Wait, wasn't this podcast about the future of biotech? Well, maybe that future requires artistic talent. Christina is indeed an expert of machine learning and network biology. She has recently started her own lab at Gladstone Institutes to study gene networks with a focus on the heart and cardiovascular disease, after completing her undergrad at Caltech, her MD PhD at UCSF, and her residency and postdoc at Harvard, the Broad and Boston Children's. But her impressive and unpredictable CV is much more than this. Christina, it's great to have you. May you describe for us your story and your passions, please?
Sure, and thank you so much for having me on your podcast. Yeah, I think first I just would like to say a bit about my passions in both art and science. I think a lot of times people think of them as separate, but in many ways they're interconnected, and the creativity that you exercise through visual art is something that's also very applicable in science as you're finding creative solutions to problem solving through the research. So in terms of my scientific passions, I started doing research in undergrad in Eric Stephenson's lab at Caltech, studying early serotonin development and the gene networks that govern that process. And I was really excited about thinking about how the understanding of the architecture of gene networks can give us insights into development and how we could apply those same concepts to human disease. And so for my MD PhD, I was at UCSF and Gladstone, where for research I was working in Deepak Srivastaba's lab, co-mentored by Katie Pollard and Benoit Bruneau, working on a series of projects developing a framework for network-based therapeutic discovery, where we first take a disease and map the gene network that's involved in the disease process so we could understand the interconnections between the genes and what's central versus peripheral. And by understanding what's central within the network, we can then design therapeutic screens to find molecules that correct the network back to the normal state, by looking at these central areas so that we can target the normalization of those and hopefully lead to more efficacious treatments, as opposed to treatments that would target something just more peripheral that might lead to more symptomatic management, but not treat the underlying core disease mechanism. And so overall, through the course of that research, we identified a molecule as that was a candidate therapy for calcific aortic valve disease, which was effective in treating cells in a dish, modeling patients from induced pluripotent stem cell disease modeling, but also was effective in an animal model at preventing and treating established disease. And we're now doing additional investigations towards clinical trial for that molecule, which is very exciting. And after my MD PhD training, I was at Boston Children's for my clinical residency in pediatric genetics, as well as my postdoc at Dana-Farber and the Broad and Shirley Liu's lab and Patrick Eleanor's lab. And there I was focusing on really targeting something that really struck me through the course of my whole prior research, which is that we really require very large amounts of data to make predictions in terms of the connections between genes within a network. And we often don't have that amount of data, especially in rare diseases and diseases affecting clinically inaccessible tissue like the heart or the large vessels. And so for my postdoc research, I developed a deep learning model called GENEFORMER, is using the approach of transfer learning so that we could learn from large-scale data and get a baseline understanding of network dynamics that we can then apply to settings with limited data and boost our predictions in those areas, even though we only have a small amount of data. And so that modeling approach is something that I'm very excited to continue pursuing and expanding in my independent work now as an investigator at Gladstone and UCSF, where we've started a group focused on using both computational and experimental approaches to map gene networks in cardiovascular disease and use that information to identify network-correcting therapies.
Thank you, Christina. That's really inspiring. And I must say that when I was at Children's, I had the luck to hear one of Christina's talks presenting this GENEFORMER architecture, and yeah, that was absolutely one of my top 5 talks I've heard during my 7 months in Boston. So, well, let's start from the beginning of what GENEFORMER is and what it does. Well, GENEFORMER uses, as Christina mentioned, a very big number of single-cell transcriptomes of around 30 million to start with, which constitute what she assembled as the gene corpus, and they come from several technologies and a broad range of tissues. Of course, they are quality filtered, and there's a lot of tricks about GPUs, software, and very interesting things that people who are in this can quickly read in her paper. And yeah, this talk will be mostly about the biology and the machine learning that's behind this great innovation. So, well, using so much data from so many studies, from so many tissues, of course, means that we have a lot of knowledge. And yeah, the key concept to start from so much data and apply it for situations where the data is not easily collectible or not so much is transfer learning on this large scale and general dataset. So, Christina, can you describe to us what is transfer learning and why it democratizes the knowledge, and what is fine-tuning to exploit and do the most of transfer learning?
Yes. So in terms of the amount of data that we have, as I mentioned, for a particular disease, we might not have a lot of data, but there has been a rapid expansion in the amount of single-cell transcriptomic data that measures the expression of all of the genes within the cell more broadly. And so we had the thought of taking advantage of all this large-scale data that's now publicly available to train a model to have a baseline understanding of network dynamics that could then improve our predictions in settings with limited data. And this is a concept that's really been widely applied in revolutionizing other informational fields, including natural language understanding, as well as computer vision, where there's models that are trained on large-scale general data sets. And then after having that general pre-training, as they refer to it, process, then we can democratize that learning to many different downstream applications. And so this is the approach that we took with GENEFORMER. And it's different than the standard modeling approach where you have a particular model that you train on labeled task-specific data to then generate a model that predicts in a particular area. But if you want to generate predictions in a new task, you'd have to completely retrain the model from scratch with new labeled data specific to that application. In this case, we're taking the model and pre-training it on these 30 million single-cell transcriptomes using a generalizable learning objective so that it gains this baseline understanding. And we can then democratize that to many different downstream applications. And so we take the model, we copy the weights for each different downstream application from the pre-trained model. And we then are able to fine-tune those models specific to each individual downstream task using limited task-specific data that would be insufficient to yield meaningful predictions if used in isolation without that general fundamental knowledge that's gained through the pre-training process. And in doing so, we're able to boost our predictions in a wide array of downstream tasks that we wouldn't have been able to with that limited data without the pre-training process.
Yeah, that's wonderful. And yeah, basically this is similar to what we as humans do in the end. We don't learn everything from the beginning when we learn a new task. We just use prior knowledge, and here this is really doing the most of all the transcriptomic knowledge that several groups in the world have accumulated in recent years to then learn new tasks when the information that you have available is pretty limited. So yeah, this was really conceptually inspiring for a student like me. So how do you present a single-cell transcriptome to your machine learning framework? This is something called rank value encoding. Can you describe for us how it is implemented, what it means, and how you came up choosing this strategy?
Yes, so for each single-cell transcriptome, it's represented as a rank value encoding. And what this entails is taking all of the genes that are expressed within that single cell, normalizing them by their non-zero median value of expression across the entire gene corpus, which we have observations of that gene's expression in many different tissues across the 30 million cells. And then after normalizing by that, ranking them in order of their normalized expression within that particular cell in that context. And so in doing so, we are able to take genes such as housekeeping genes that are ubiquitously highly expressed and normalize them by a larger number such that they move lower in the rank value encoding or deprioritized by the model. Whereas transcription factors, which we know are really important for regulating the expression of other genes within cells, these are often very lowly expressed, but when they are expressed, uniquely distinguish cell state. So through the normalization process, we're then moving them higher within the rank value encoding and prioritizing them for the model. Additionally, the rank value encoding approach is one that may be more robust to technical batch-dependent artifacts, whereas the particular expression value might change and be more sensitive to batch-dependent artifacts. The relative ranks may be more robust across these different settings. And we did indeed find that batch-dependent artifacts were the model was robust to batch-dependent artifacts using a variety of different types of artifacts that are commonly perturbing the data, including preservation or platform used for the librarian sequencing or individual patient values. And so that's another benefit of using this type of approach. Additionally, one thing that is also important to consider is that the type of model that we're using is an attention-based model, and we can talk a little bit more about the attention mechanism and how that works. But generally, the attention mechanism is something that's used for sequence-based data. And so there's been a lot of efforts broadly in many different informational fields to arrange data such that it can be represented as a sequence and therefore utilize this powerful machine learning approach. For example, taking images and representing them as a sequence, as an array across the image, or movie data, sound data, etc. In this case, we have used a rank value encoding and used that as the input for an attention-based model, indicating that this is potentially something that can be used in any informational field or type of data that can be represented as rank values. And therefore, that really expands the possible utility of this modeling approach to many additional types of data.
Yeah, I'm wondering if this is more of a machine learning paper or a biology paper. Probably it's really advancing both fields at once. So that's also inspiring for students like me. And so to sum up for maybe people who are less familiar with these topics, for ones of you that may have some different background, basically what we have discussed so far is people in several labs in the world have produced tons and tons of data of single-cell transcriptomes. But now, how do you use these single-cell transcriptomes, which in the simplified form that we're discussing in an outreach attempt are basically matrices counting for each cell the level of expression of each gene in the genome? How do you go from these matrices, let's say, the very simplified idea that I want to convey, how do we go from this to a machine learning that can really use them to make what Christina was describing. We use this rank value encoding, which has lots of advantages in a number of fields that Christina has described greatly. And then there is some well-tuned mechanism to use this data, which is based on self-attention, as she mentioned. So now, can you please describe for us, Christina, what is self-attention and what is a transformer encoder unit, which is the basic unit of GENEFORMER, please?
Yes, so GENEFORMER is composed of 6 layers of transformer encoder units, and this type of machine learning architecture was developed primarily initially in the field of natural language understanding for language modeling, in several papers, including Vaswani et al., which is the paper called "Attention is All You Need," that I strongly encourage everyone to read to get a stronger and deeper understanding of this modeling approach. But the basic composition of the transformer encoder unit includes a self-attention layer and a feed-forward neural network layer. And the self-attention layer is perhaps the more interesting of those two. And so for the self-attention layer, the major benefit of using attention is that you're able to have a model that can take into account very large input spaces and learn which areas within that large input space are important to focus on to optimize the particular learning objective at hand. And generally speaking, in this case, when we're looking at genes that are expressed within cells, the model is learning which genes pay attention to which other genes within the cell and vice versa to optimize the particular learning objective and gain the prediction that you're asking the model to learn. And this is something that is optimized through the course of the training process with giving feedback to the model in terms of its predictions, whether they were correct or not, and fine-tuning these weights within the model to optimize which genes are paying attention to which other ones. And the benefit of using this approach, and in this case, is that we're able to take into account the context of the other genes within the cell to make predictions on the identity or function of particular genes or that cell as a whole. And the very nice thing about this model is that it is therefore context aware. So when we make predictions, we're making predictions specific to that particular cell's context, and therefore we're able to get much more subtle information about genes or about cell states than we would in terms of a modeling approach that's not context aware, where we're making a single prediction for a gene across all cells, let's say, which we know genes act differently in different contexts. So in this case, we're able to take advantage of that information.
So yeah, to strongly simplify this last concept, well, a neuron and a red blood cell, of course, will have differently wired gene networks, and their model can keep this into account. So for instance, if a gene is regulating a gene, let's say gene A is regulating gene B, in neurons, it's not so obvious that the same regulation is happening in blood cells, let's say. So we want to have a model that can predict if the absence, let's say, for instance, of gene A in neurons is causing dramatic effects on gene B. But if we have a very general cell type, maybe this is averaged out, but in neurons it's strong and in blood is very faint, or the other way around. And so, yeah, we can keep into account different cell types, of course, because all the data that are aggregated do keep into account the cell type of origin of that single cell transcriptome, and that's really inspiring. So maybe this gives us the opportunity to introduce what gene and cell embeddings are that you greatly describe in the paper. And I think they are like the basis for context dependence, right?
Yes. And to speak a little bit more about the context as well, it's also really important for disease modeling as another example to the one that you very correctly put. And so, for example, we have genes that we know are expressed across all tissues in the body. But when we lose a copy of those through a particular mutation in patients, we get a really specific disease that affects a particular tissue and at a particular time point within the course of their lifetime. It could be something during the course of early development or something that develops with age later in life. And so we know that the context is really important in understanding where the effect of losing that gene or decreasing the dosage of that gene will play a role in developing the disease process. And so we use both the gene and cell embeddings to help us understand that on a context-specific level. So the model has the outputs of both gene and cell embeddings. So when we say embeddings, we mean that the model is generating an embedding space, which is in this case a 256-dimensional space. And those 256 dimensions, the model has learned through the course of the training to have particular attributes of genes or cells within that space on those different dimensions. And so when we map particular genes within that space, we can see that genes that we know have similar attributes across some of these dimensions will be closer together within that embedding space, whereas genes that have very different functions will be markedly separated within that embedding space. And so essentially, it's kind of a map that the model constructs through the course of the training process to position each of the genes across this space so that the model can understand for the particular prediction that we're asking for where these particular genes lie for the attributes that are important for that prediction. And so to give a more concrete example, so for example, when we're thinking about a particular downstream application of understanding whether decreasing the dosage of a gene will be detrimental to that particular cell, we ask the model, we fine-tune the model by giving it a few examples of these types of genes that are detrimental, and then asking it to predict that for other genes is one way that you're able to do this. And so through the course of that fine-tuning, it will separate within the embedding space the genes that are detrimental versus those that will not have an effect. And so when it sees a new gene, as it runs through the model, it's embedded in a position within that space, and you can see, is it close to the genes that are detrimental or close to the genes that are not? And therefore, the model can help predict and interpret what the effect of that gene's decreased dosage would be. In terms of the cell embedding, that's an aggregate of all of the genes within the cells, their embeddings. So we take each gene's individual embeddings and then we aggregate those together to get the cell in general, its embedding based on the genes that are expressed there. And so similarly, cells will be embedded within the space separately depending on their cell state. So if we train a model to distinguish between cells that are from hearts that are healthy or non-failing, from cells that are from hearts that have particular cardiovascular disease like hypertrophic cardiomyopathy or dilated cardiomyopathy, we can then identify where the cells lie within the embedding, and they will be separated based on their cell state of being healthy versus from one disease state or the other disease state. And the helpful thing about having these cells separately embedded within the space is that we can then start to do experiments to say what is really contributing to cells being embedded in that particular position. So if we take the cells that are from healthy hearts and we start in silico or through the computational approach, deleting particular genes, can we see which genes deleted in the normal cells will shift their embedding towards the hypertrophic cardiomyopathy disease state or the dilated cardiomyopathy disease state, because that will give us an understanding of which genes are really important in defining these disease states and contributing to the disease process. Or alternatively, if you start from the hypertrophic disease state, let's say, can you delete particular genes in silico through the computational approach and see then whether these cells are now shifting back towards the embedding in the normal state, while also shifting away from the other dilated cardiomyopathy state, so we don't have potential side effects from candidate therapeutic targeting. And so there, we can use that approach as an in silico treatment strategy to identify which genes are really important for defining that hypertrophic cardiomyopathy state, and if targeted, will move the state back towards normal and therefore could be promising targets for therapeutics. And so those are some of the ways that we can use the gene embeddings and cell embeddings to help us understand the underlying mechanisms in the disease process or other types of cell function, and use this context-aware modeling approach to identify the predictions that we're interested in.
Okay, that's wonderful. So to oversimplify again, this in silico deletion approach is mimicking something that we as biologists have been doing for lots of years, which is deleting a copy or two copies of a gene from a cell and see what happens. And this can be useful first to probe the function of the gene, because if the gene is not there, then what happens? I mean, is the cell behaving differently? So maybe that can point to the function of the gene. On the other side, it can be useful to block a gene function for therapeutic purpose, or on the opposite spectrum, you can hope to upregulate that gene to increase that gene expression, so to compensate for some function. Or if you want to study a disease, maybe in that disease that gene is not working, so you want to block it and see how the cell reacts to the lack of this gene. And it's really incredible to me that from this ton of data and this clever machine learning, we can simulate this with great detail, and I'm really looking forward to the applications in the next few years. And to connect with this, there's one suggested application in the paper which I really liked. That is, Christina and colleagues suggested that this in silico deletion approach can support GWAS variants prioritization to point at putative causal variants. What I mean is genome-wide association studies, or GWAS, are correlative studies that try to link phenotypes and the underlying genetic variants. But as they are correlative, they are pretty hard to interpret and amenable to false positives. So having an in silico deletion approach means that you can try to see what happens in the gene network, how the gene network rewires, if it's switching to a network more similar to a healthy phenotype or disease phenotype, and what is happening in some areas of this network if you delete this gene. So that can really point to some putative causal action that you can then inspect experimentally, probably. So can you, Christina, describe for us what this means, and how did you come up with this idea, and what are the next steps you want to do on this?
Yes, so I think in general, for one, prioritizing GWAS, but also in general thinking about what are the important genes that are involved in disease processes. And when we perform this in silico deletion, where we artificially reduce the amount of that gene that is within the cell in our model and see the effects of that, can we understand what is the effect expected in all of the different cell types or different developmental time points within the tissues? And in doing so, we can get a context-specific comprehensive map of each gene when downregulated through a particular mutation. Let's say, what is the effect that we expect in terms of the phenotypic spectrum in patients? In which tissues is it affected? In which developmental time point do we expect? And we can also do this in combination. So another important concept is understanding why a particular gene, if we have the same mutation, even sometimes within the same family, we see different manifestations within patients, even within that same family, where some patients might be more mildly affected, whereas others are more severely affected in the disease. And could it be that there's additional mutations in other genes or regulatory regions that are impacting the expression of that primary mutation? Or could there be multiple mutations acting together to affect the severity of the disease? And so we can also, in silico, be able to test this in combinations. So for particular genes, let's say that have a low effect when we reduce their dosage in silico, in combination with other genes, do they now have a strong effect? And in doing so, we're able to prioritize these potential combinations computationally, perhaps more efficiently than would be possible with experiments. Because usually with experiments, we're really following up on the strongest effect targets that we see within screens, and it's not feasible really to look at all the different combinations, whereas we could do so more efficient computationally.
That's really inspiring and impressive too. So going back to the machine learning that's empowering all this before delving deeper into all the applications. We've described in detail how the single-cell transcriptomes are encoded and what are the self-attention transformer encoder units. But how is the learning objective? You described a masked learning objective, which was new to me, but it's really described well in the paper. And it is really building the self-supervised mechanism that enables this to perform greatly without the use of any label. So can you describe how it works for us, please?
Yes, so the strategy for the learning objective is distinct within the pre-training phase and then the fine-tuning application specific to each downstream task. So in the pre-training phase, your goal is to have a generalizable learning objective, something that will teach the model general and fundamental knowledge about the learning domain, in this case being network dynamics within cells. And then in the downstream applications, you can have a much more specific learning objective to that particular downstream task to fine-tune the model to gain predictions in that particular area. So for the pre-training objective, that's where we use the masked learning objective. And this is something that's been shown to be very effective and generalizable in other informational fields as well, such as natural language understanding. And within the masked learning objective, what you're introducing to the model is the rank value encoding with some of the genes, in this case 15% of the genes, masked. And so within that rank value encoding, 15% of the genes will be masked or blocked such that the model cannot see what they are. And you ask the model to predict, based on the context of all the remaining unmasked genes in that particular cell, what is the gene located in that particular position within the rank value encoding in that cell. And in doing so, you can have the model learn as a general idea when certain genes are moving up or down within the rank value encoding, where are the others moving, and therefore how are they interconnected? Do you see an impact of particular genes on the position of other genes within the rank value encoding? Are some just stable and not really being affected by that particular change? And that is the objective we used for the pre-training process. The other major benefit is, as you said, it's completely self-supervised. So with the pre-training corpus, we can use all of these 30 million cells, even without any accompanying label data, and instead of restricting ourselves to only data that includes labels. This allows us to take advantage of much larger scale data within the pre-training process. And then for the fine-tuning stage, then you can use label data, particular for that downstream task and a particular learning objective. For example, distinguishing states of cells, either normal or disease cells, or states of genes, function of genes, such as genes that are likely to be dosage sensitive. If you decrease their dosage, lead to a disease versus not.
Wonderful. So yeah, let's jump to these downstream applications. We have already discussed briefly what one can do as for in silico deletion, or in general to fine-tune the dosage sensitive genes in order to see what happens if you increase a lot or you decrease a lot the quantity of a gene, and to do so in a context aware way, so to probe the effects of the dosage of each gene and combinations of genes in different cell types or developmental time or patients. So with great detail, and I don't think there's any model out there capable to do this in such a detailed way. And our application that I wanted to quickly introduce is the in silico reprogramming of fibroblasts. So we as biologists do like to switch back the skin fibroblasts to cells that are similar to the embryonic stem cells. They are called induced pluripotent stem cells, and they are one of the protagonists of this podcast. And the idea is that by treating with the so-called Yamanaka factors, the skin fibroblast, one can induce a state which is pluripotent, so it can really form basically all the tissues. And the great thing that you can do with GENEFORMER is you can just put the Yamanaka factors at the front of a rank value encoding. And by just doing something like this, which is much simpler than adding to a dish all the viruses and doing all the experiments and see if it never works and try it again and everything, you can see that the other genes in the transcriptome do shift their embedding to a state which is much more similar to the IPS. This is evaluated by a measure called cosine similarity. You can think that, as Christina described, cells and genes are embedded into spaces which are highly dimensional, and you can see like the projection of two different arrows one to another with the cosine, and this is something like that. So very oversimplifying, of course. This really proves that the Yamanaka factors also in this model can really shift a transcriptome from a well-differentiated cell type such as a fibroblast to a transcriptome which really seems pluripotent. And similar to this, they also performed in silico differentiation by using MyoD, which is a known differentiation factor. So what we think is that the artificial neural network does have an understanding of the gene network. It's not really, I mean, it's quite impressive that it can really understand and perform such at great detail experiments as we do in the lab. So another application that I really loved and I want to discuss with you, Christina, is chromatin dynamics prediction. So can you describe to us what is this task and how you implemented it?
Yes, and for the reprogramming or differentiation, as you mentioned, we're modeling the effect of adding these particular factors and whether it shifts the cell. We're only modeling the first step of that process. So, we see a particular shift in the remainder of the genes, and that tells us that the model is context aware. So those genes embeddings are shifting towards what they would be within the pluripotent state or, the end state of that differentiation. But in that case, we're just modeling the first step of adding those, and, all the other genes were keeping the same. But you can envision a future application where you're able to model each step and then see the next step and model the next step from there, kind of similar to a next sentence prediction type of modeling approach that's used for natural language understanding, to be able to make better predictions of how to direct cells from one cell state to another. So I think that's something that could be really interesting for a future application. And then in terms of the chromatin dynamics, so in this case, we were, again, as a proof of concept, attempting to test the model for a wide variety of diverse downstream applications. And in this case, we tested chromatin dynamics for bivalently marked genes. And so there's particular genes that are known to be bivalently marked at their promoters or the initiation site for their expression. And these bivalent marks are both a mark of activation and a mark of repression on the same gene. And it's been shown previously, initially in Bernstein et al. In 2006, that these particular genes are known to be developmentally important when we see these types of marks. And so we wanted to test whether the model, if given a few examples of these bivalently marked genes, could predict these bivalent marks on other genes. For example, if the gene is single methylation marked or bivalent marked, or not methylated at all or bivalent marked. And so to accomplish this, we first trained the model using just the initial bivalently marked genes that were reported in that initial paper, Bernstein et al. 2006, where they just looked at 56 highly conserved loci. And in doing so, we were able to train the model with an approach where you look at 80% of those and then predict on the remaining 20% and found that it was effective in learning that for these highly conserved regions. But then what we did was training the model just on these 56 highly conserved regions. We wanted to test whether then the model could predict genome-wide in the data from later studies where they were able to look with genome-wide approaches to identify bivalently marked genes across the entire genome, not just these 56 highly conserved loci. And we found that the model was able to gain meaningful predictions genome-wide and boost the predictions compared to alternative approaches using just this training on the 56 highly conserved loci. And so that was something that was very interesting to us because we're only using transcriptomic data as the input to the model, but it's able to gain some predictions in terms of chromatin dynamics on these particular genes in terms of the bivalent marks. And it's not just their particular expression in embryonic stem cells, which is the cell types that from the data that we were training on, because if you look at alternative approaches, they're not able to gain those strong predictions. So it's likely the fact that the model has seen these particular genes in the many different cell types used from the pre-training process and been able to recognize a particular pattern within the embedding space that indicates some level of encoding of this type of chromatin information that then would downstream affect the transcription and be reflected in the transcriptomes that we're looking at.
I'm impressed by how GENEFORMER really understands molecular biology in the nucleus much more than me. Wow. So yeah, maybe we can mention another application before jumping to more speculative questions that I'd love to ask you. The last application I'd be really happy to discuss is how you can normalize co-regulatory elements in disease settings and perform what you called in the paper in silico treatment to discover candidate therapeutic targets. For instance, you have examples in cardiomyopathy. So you performed a specific fine-tuning. How did you do it? How did you confirm it experimentally? So what's the general value of all these for pharmaceutical design in the years to come? Thank you.
Yes. So for the in silico deletion approach, the general idea is understanding when you delete a particular gene, what is the effect on the remaining genes within the cell, and then through an aggregate of those, what does that entail for the effect on the cell as a whole? And so actually just taking the first step of that and understanding when you delete a particular gene, what do you see as the effect on the remaining genes? We were able to see that, for example, when we look at cardiomyocytes, where we know that there's particular transcription factors like GATA4 that are really important for cardiomyocyte function, when we delete in silico GATA4, we see that the strongest effect is on the genes that we know are directly downstream of GATA4 based on data that we have from experiments that look at where GATA4 binds on the genome and which genes are impacted by reduction in its dosage. And these are the most strongly impacted compared to indirect targets or targets of other transcription factors or housekeeping genes, which are stably expressed and not expected to be affected by in silico deletion of this transcription factor. And so this tells us that GENEFORMER is able to understand the hierarchy within the network in terms of what is the most directly impacted by this in silico deletion. And we also found in other applications that GENEFORMER was able to encode within the attention weights the relative importance in terms of what was most highly attended to from genes that were central with the network versus peripheral. And so I think that understanding of hierarchy is really important in terms of its predictions within disease settings, because like we discussed earlier, when we are able to understand what's central within the network, we can really target the core disease mechanism as opposed to something that's just peripheral and may lead to more symptomatic management. And so in terms of the in silico treatment approach, so when we have the model distinguish through the fine-tuning process between cells from normal, healthy, or non-failing hearts compared to hearts that are affected by hypertrophic cardiomyopathy or dilated cardiomyopathy, that fine-tuning process of teaching them all to distinguish between these pushes the embeddings of each of these cell states separately within the embedding space. And then once you have defined where these cells fall within the embedding space, you can do this in silico deletion or in silico activation of genes to understand whether we can take cells that are from a particular disease state, let's say in this case dilated cardiomyopathy, and push their embeddings back towards the normal, which is what you would want from a therapeutic approach. And so we also want to push it towards normal, but also away from the other opposing disease state of hypertrophic cardiomyopathy so that you don't generate potential side effects. And so we were able to get candidate therapeutic target predictions through this approach. And we actually now have tested some of these within cells that are modeling dilated cardiomyopathy in the dish experimentally. And we have found that there is concordance with the predictions and what we see in terms of genes that, when targeted experimentally, are able to actually improve the force generation of the heart muscle cells back to what you would see in the normal healthy setting as opposed to the disease setting. And so that gives us additional support of using GENEFORMER as a tool for discovering candidate therapeutic targets, and additionally, several targets to follow up on for dilated cardiomyopathy, and additionally, we hope to pursue the hypertrophic cardiomyopathy case as well as potential candidate therapeutic targets that we can design therapies to help the patients suffering from these disorders where there's no targeted medical treatment currently.
That's really exciting. And I was thinking while you were talking that you also have an approach to predict holistically also toxicities of deleting or increasing the expression of a given gene as a pharmaceutical intervention, which is to me a lot, because typically what I've been reading only examines toxicity at a very, not holistic way. You really have a hard time understanding the effects on a whole cell unless you go sequencing it for real. That costs money and effort and everything. So yeah, I think that this is great not only to test the efficacy of potential pharmaceutical interventions, but also to predict their possible toxicities by mapping onto the gene network.
Yes, and I think that this would be really great to apply in the future to additional diseases, both in cardiovascular disease and in collaboration with others in other disease states. And yes, there is potential applications too for thinking about, as you mentioned, toxicity and prioritizing drug screening from this.
Yeah. So now, Christina, I have very many questions which are more speculative, but I'd really like to ask you because there are so many potential applications and downstream inventions relating to your project, I think. So I'm very curious. And well, first, I want to ask you if you think that in the near or less near future we'll be able to account for isoform level and or allele specific transcripts in the ranking to model, for instance, transcriptome-wide effects of splice altering mutations or the inactivation of a single copy of a gene that may result in haploinsufficiency. Well, maybe here I have to define a couple words. So, as many of you may know, genes are transcribed into RNAs, and these RNAs are not what is translated into protein, that is the machines of a cell. But first there is splicing and several other cellular mechanisms that ensure that from the transcript deriving from a gene, you have something that you can really translate into protein. And this splicing is something that has alternatives, so it's called alternative splicing, and you can really split things and paste things together. So different copies of the same gene will result in a number of different transcripts. And allele specificity instead refers to the idea that you have one mom copy and one dead copy for all of your genes but your Y genes, let's say. So, and X genes for males, of course. So can you keep into account this allele specificity? And the reason to ask this is that many diseases are heterozygous, meaning that the mutation impairs only one of the two copies of a given gene, and this is the case for the haploinsufficiencies, which I mentioned, in which a single working copy of a gene is not enough for proper function. So Christina's description of GENEFORMER suggests the in silico deletion, but I suppose this is complete in silico deletion. Of course, you can model haploinsufficiency possibly by changing the ranking of a given gene, so it's kind of halved its expression, but I want to ask this in detail. So do you think we can really account for isoform level or allele specificity to model this plethora of disease-related mutations?
Yes, that's a great question. So as you mentioned, in this case, we were modeling, either completely removing the gene from the ring filling encoding or adding it to the very front of the ring filling encoding in terms of the activation. And so there's certainly more subtle changes that you could make. In terms of the haploinsufficiency, I think, we would have to look at cells that have one copy missing of the two for a particular gene and see where the gene falls in the ring filling encoding, because it could be that it falls far enough below the threshold where there's dropout that actually completely removing it is modeling similarly haploinsufficiency and complete deletion. But you can slide the genes up and down the ring filling encoding in a more subtle way. So, for example, you could try to model 25% increase in that particular gene's expression by taking all of the cells and where you see it within the ring filling encoding, moving it, some degree of that's reflective 25% up within the ring filling encoding. And so I think that there's definitely more subtle in silico perturbations that can be taken to further examine that. In terms of splicing and isoform specific effects, because the model doesn't start from sequence to get to the transcription, it would be, I think the approach to do that would be to connect that with some of the tools that you have, for example, Splice AI, where it can kind of model the impact of particular splicing variants. And you could have, I could envision a setting where you could combine that with a tool that starts from the sequence to predict what you expect in terms of the expression, and then use that to model what that change in the ring filling encoding would be as an input to GENEFORMER.
Yeah, that's exciting. I was also thinking that maybe in some single-cell transcriptomes, depending on the technology, there are tools that can predict at least partly, and this is technical, but partly, let's say, the splicing that's happening for each gene. So maybe there could be some model like GENEFORMER that starts from isoforms instead of gene-level counts. So you just use another software in the very pre-processing phase to make gene counts instead of, to make transcript counts instead of gene counts, to keep into account splicing isoforms. I don't know.
Yes, definitely. I think there could be another step in the beginning in terms of generating that initial ring filling encoding that includes an embedding of those more specific isoform effects.
Nice. So connecting to this, I want to ask if you foresee developments to make GENEFORMER the basis to create so-called artificial patients. What I mean is, if we assume that the transcriptional network is fundamentally wired in the same way in all of us, but our unique genomic variants make us genetically unique indeed, can we have someday an algorithm that understands how these variants impact the network for each cell type, as we've discussed context specificity? So, well, if we were able to make such a pre-trained model to be then trained with patient-specific genome, we may be able to understand if that patient's transcriptome substantially deviates from a populational average in a given cell type. For instance, if their heart has a transcriptome that is more similar to that of a failing heart than non-failing heart. So I see huge implications here because, by just sequencing patient's genome, we may be able to strongly predict these predispositions, even without maybe variant calling, and orient corrective lifestyle or medicines that are predicted by the algorithm to bring back the susceptible cell type, as Christina was describing, to a transcriptome that is more similar to healthy individuals. And I come from the world of rare diseases, so I also think that this may be really valuable to formulate hypotheses for idiopathic conditions. And what I mean by idiopathic is conditions that don't have a known cause. There are several rare diseases where we don't know the genetic cause or environmental cause. We just know that the phenotypes are very remarkably similar. So maybe we have a patient with epilepsy, let's say, and a few putative gene variants. Okay, we found them from the sequencing of its genome, of their genomes. So we want to prioritize these variants. We run some algorithms similar to what Christina has described for us, with combinations of these variants in case we can keep into account the variants from different patients to then narrow down which of these variants may be causing the disease phenotype. For instance, shifting the given neural cell type that I was describing to a transcriptome that is more similar of other diseases that has this epileptic phenotype, for example. So yeah, I'd love to hear your thoughts and comments about these possibilities. And I think I've talked a lot, so I will repeat the initial question that gave rise to all this monologue. So do you foresee developments of GENEFORMER to make this the basis to create artificial patients and jump from genome sequencing to predicting specific cell type specific misfunctions in a given patient genome?
Yes, no, that's a really great question. And I think for what I was describing before in terms of in combination, silico modulating multiple genes, that's one thing that we're able to do with GENEFORMER currently that I think is really impactful in terms of understanding how many genes might work together to lead to a disease process. Or in the reverse, if there are particular genes that are actually protective, and could there be variants in patients that are actually protective against the disease process from progressing? That also leads to some of the variability we see in different patients for the manifestation of the disease and its severity. In terms of coming from the sequence level and using that as the basis for predicting transcriptional changes, I think that also comes into the idea of using other data sources besides transcriptomes as corollary information that can be used by the model to better understand the cell states and the gene's function. And so I think in the future, as there's more data that's available, linking those to genome sequencing and transcriptional studies in different cell types or time points, or additionally other types of data like chromatin accessibility, where we can see open regions within the genome that can give us an understanding of where the important elements are that regulate the expression of downstream genes. Using many different data types like that in combination as a multitask learning approach can potentially also help to improve the predictions that we were getting and model patients on an individual way that includes their particular variants as you were describing. And so definitely I foresee in the future opportunities to integrate more data types to improve our predictions.
What a time to be alive. So, well, I see that GENEFORMER is much more clever than me, so I want to ask how we can probe this intelligence. I think that GENEFORMER is learning genomics at a very deep level to understand something of regulation that maybe also entails the protein and functional level, even if it has never seen this data. And I was also struck by the example that previously Christina briefly mentioned of in silico deletion of GETA4. GENEFORMER is indeed able to retrieve the well-known altered downstream targets, but the effects of the lack of GETA4 are manifest at the protein level, so it seems to me that, at a first sight at least, GENEFORMER can understand something that is more than a correlative gene network. It understands causality and at a deeper level that gene expression. So, well, I am having fantasies, I must admit, to interface such understanding with recent advances in let's say, predicting the impact of mutations on protein structures. Indeed, a promising and idea is propagating a missense mutation down to the affected targeted interactions, and this may help us understand more of what amino acid substitutions can do, and especially what are the rules that govern gain of function versus loss of function. So, there are mutations that create novel functions in proteins that may be deleterious and lead to disease because something new is happening in the cell which should not be happening in a healthy context. Or on the other hand, some mutations might impair protein function, so it is as if that protein wasn't there. So loss of function might be another case, but it's always very hard in the lab to discriminate between the gain of function and the loss of function of a novel mutation, especially in the rare disease setting. So how can we use GENEFORMER to understand all of this? And do you really believe that its understanding is going much deeper than the gene expression level, and how maybe?
Yes, no, I think there's definitely a lot of research in general into understanding how attention-based models are able to use the input data to gain predictions in many informational fields, and it's definitely an active field of research. I think for GENEFORMER specifically, we looked at the attention weights to understand whether through the pre-training process, before any fine-tuning, could the model have learned particular aspects of genes that we know are important for regulating other genes. And we did find that for particular attention heads, they significantly attended to transcription factors, which we know are important in regulating other genes compared to other genes within the genome. And so in a self-supervised way, the model did learn the relative importance of transcription factors without any prior information or labeling of those. And additionally, we found that particular attention heads paid higher attention to central genes within a defined network compared to peripheral genes, or more highly ranked genes within the rank value encoding that are, because each rank value encoding is unique to particular cell states in terms of what's most likely to distinguish the cell state and therefore be higher in the rank value encoding. So there is encoding of the network dynamics that happens through the self-supervised process of the pre-training that is really interesting to probe further. And, I think additionally taking a look in the future also at particular fine-tuning applications, how those weights are updated to generate the predictions specific to that task. The embeddings themselves are also important to consider in these cases because in some ways the embeddings can be thought of as an aggregate of all of the attention weights and the layers that came before that embedding, and that embedding is now encoding the information important for that prediction. So I think there's definitely a lot of room for future research and understanding how transformer models work in general, and also specifically in this case with regard to this biological application.
Yeah, so what about the possibility to interface GENEFORMER and other software emerging in the field of protein interactions to study, for instance, the difference between gain of function and loss of function at molecular level and understand it at a holistic cellular level, maybe?
Yes, I think that, similar to what we were discussing before in terms of the chromatin accessibility or genome sequencing data, protein level data is also something that would be very exciting to integrate as well and give information to the model about the predictions for particular changes that we might expect to change a structure and lead to a gain of function or a loss of function based on changes to particular binding domains for proteins or complexes. And understanding also those complexes could be informative for understanding how combinations of different genes work together to lead to a particular disease process or to be protective against a disease, as well as understanding how particular downstream targets have differential sensitivity to reduced dosage of their regulator. So when you have reduced dosage of that regulator, there are certain downstream targets that are going to be more sensitive and more highly impacted compared to others that are not going to be as highly impacted. And so what really underlies that a context-specific sensitivity to a particular haploinsufficiency or reduced dosage of a regulator. So I think all of these types of data, including protein structure information, could be very interesting to integrate in future approaches.
So guys, make sure to follow Christina's work in the next few years because exciting things will happen. So now I have a very edge question, I think. Do you believe that GENEFORMER's thought, let's call it, way of thinking, I don't know, may learn patterns that explain some of the underlying biology of gene regulation that may be novel to us.
Yeah, I do think that, again, by looking at kind of the underlying structure of the attention weights and other aspects in the model and how the model is making these predictions could yield some important information. But additionally, through these downstream applications, if there's a particular area where we want to understand more, we can fine-tune the model using particular task-specific data and gain better predictions, or use these in silico perturbation approaches to gain a better understanding of the effect of particular genes. And I think that's something that would definitely be very exciting to be able to apply the model to understanding and making predictions that we can then test experimentally to understand, for example, what is the underlying mechanisms of context specificity of transcription factors, like we talked about, in different settings or at different individual loci within the genome, which is something that is not well understood, and something where we have, I think, a great opportunity to gain advances in through this computational approach.
Wow, yeah. And as we work towards then, I want to ask if you plan to make tutorials for researchers to use this incredible resource, because it sounds so great that it's maybe intimidating. What do you plan to do to make it greatly available to everyone?
Yes, definitely. The model and the pre-training corpus will all be freely available and open source, with additionally examples and, as you mentioned, tutorials to walk through the different steps of using the model for some of the main applications we discussed within the manuscript. And we hope that by providing this information, it can help to facilitate other groups using the model for their predictions, and also in collaboration, working with others to apply this to additional disease settings, and hopefully advance both our basic biological mechanism understanding and disease modeling with this pre-trained model as a basis.
Sure. So where do you see this research project in, let's say, 5 or 20 years from now? This is a hard one, I know.
Well, I think, as we discussed, some of the very interesting applications are both for disease modeling and understanding some basic mechanisms of biology, like the context specificity of transcription factors in different settings, and whether we can understand the transcriptional and epigenetic mechanisms that underlie that. We hope that also there's additional modifications and additional modeling approaches that we integrate, including using this model for a more explicit network inference approach, where we can really map directly the connections between genes, as well as integrating additional data types, like you mentioned, with chromatin accessibility. Right now, there's some small amount of multi-omic data available with transcriptional and chromatin accessibility information in the same single cells. But we hope that this, over the course of the next a decade or maybe sooner, will really expand and give us enough data that we can generate a pre-trained model that incorporates that information and help us understand the regulatory elements important for particular diseases. I think the other kind of main goal of the lab is synchronously advancing computational approaches with a lot of the new biotechnology advances that are happening. So I think as we gain new biotechnology tools that enable us to measure aspects of cells that we could never see before, we now have the opportunity to also generate advances in the computational analysis of that new data to gain the best insights as we can to the biology and disease modeling through those approaches. So in general, kind of more broadly, that's another major goal of the lab. And hopefully, we, in the next set of years, as you mentioned, are able to see the outcome of some of these testings that we're doing with the candidate therapeutic targets, and hopefully be able to develop a targeted therapeutic for these cardiovascular diseases that currently have no medical therapy and are progressive over time, where we have a really great opportunity to intervene. That's the kind of end goal of all of the work, is to bring this back to the patients and help improve their disease process.
I'm really looking forward to it. So, quick question, maybe not so quick, what are the major obstacles that you think you will have to deal with on the computational or wet lab sides?
Yeah, so I think one thing computationally that will be really important is, right now, the amount of data that we see from single cell transcriptional sequencing is a certain number of genes are being detected. But in the future, we hope that the technologies will advance, or sequencing will become less expensive, and this will be able to be sequenced deeper, and we can detect more and more genes. And so one computational aspect that's important to consider is that we use a full attention mechanism, where all of the genes are being trained in terms of their weights of which other genes to pay attention across the entire input space. But if we look at how this impacts the speed of the computational algorithm, it has an effect that is quadratic in terms of its increase and how slow the algorithm becomes. And so there have been a lot of advances in primarily natural language understanding, but also other informational fields, to generate approaches for sparse attention, where you can estimate the attention by not having every single point within the input space pay attention or learn to which genes to pay attention to in the entire cell, but take a more sparse approach, such that then you can be a lot more computationally efficient in terms of being able to take into account much larger input spaces. So I think that's something where there's definitely room for algorithmic advancement as well in application in this biological model. And then experimentally, I think we are continuing to gain additional types of biotechnological tools in terms of the way that we can manipulate cells to model particular variants, let's say, or diseases, so that we can improve our modeling in the dish, and therefore be able to get a better prediction of therapeutics that could be effective when we're looking at both the in vivo setting that's three-dimensional, has many different cell types involved, and hopefully also translate that to greater efficacy in patients for clinical trials. So I think with over the course of the coming years, advances in both the computational and experimental domains will highly drive the progress in this field.
Okay, wonderful. One last question. I mean, this time I'm serious, it's the last. What do you dream to achieve thanks to this incredible work relating to your research focus, which is in cardiovascular health? So a dream of yours to close with.
Yes, I think for me, as because I also see patients in pediatric cardiovascular disease, it's something that to me is the main goal and motivation of all of my work, is to identify targeted therapeutics for these patients that are suffering from these progressive diseases where we really have the impact to intervene. So for me, the dream would be to, having started from the patients and modeled the disease and understood potential candidate therapeutic targets, the dream would be to develop a targeted therapeutic that then we do see efficacy in clinical trial and is able to be brought back to the bedside to these patients. And that is kind of the main motivation of all of the work in the lab, and what I would see as the dream of the outcome of all of this work.
That's absolutely a dream worth pursuing. So thank you so much, Christina. This has been a wonderful conversation. Thank you for your time.
Thank you so much. Really appreciate it.
You've just listened to A Biotech Futurist, a podcast by Luca Fusar-Bassini. This is the first series, and a new episode is out every Monday. Please consider subscribing and rating the podcast on Spotify, Apple Podcast, YouTube, Instagram, or your top podcasting platform. And if you liked this episode, consider sharing it with your friends, as the growth of new podcasts relies on word of mouth. If you have any suggestions, don't hesitate to reach out to me on Instagram or Gmail at thebiotechfuturistpodcast@gmail.com. You can find the full AI-generated transcript of this episode on my website, lucafuzarabassini.com. I'll also post the links to the main papers referenced in this episode, which you can find here in the description too. Thanks for listening to A Biotech Futurist. I am looking forward to talking with you in a week.